Hello, everyone. I'm Inat. I'm co-founder at Emedgene. And before I begin, I want to thank the AWS team for arranging this fantastic event. It's really great to be here. So a quick intro about Emedgene. Emedgene deciphers how patients' genome impact their health. And since we're doing so using technology, we're able to scale genomics-driven care to mass populations. I don't know how many of us realize this, but healthcare is in the midst of a major leap towards genomic-informed care, which means that as patients, our genomic data is going to be regularly used for clinical decision-making. Now, this is not a leap of faith anymore. This change is already underway, and it is happening at an accelerating pace. For some of us, it's already a common truth because we're seeing the convergence of trends that enables it to happen. But we have a mixed audience here today. So I'm going to take the next few moments to describe what we innovators view as tectonic shift in this market and why it should matter to you whether you're a clinician, a hospital CIO, or a pharma R&D manager. This change is coming to you too. So reiterating here some of the major trends that are accelerating the adoption of genomics by mainstream healthcare organizations. Over the last decade, there's been a 20-fold reduction in sequencing prices, where NGS stands for next generation sequencing. And that drove more research that in return brought proof of clinical utility and outcome, and as a result also improved reimbursement. So there is a continuous growth in the number of lives insured for advanced genetic tests, with over 50% of the US population now covered for exome sequencing, which is a test that looks at patients' full spectrum of genes. And with the improved reimbursement trends, a proliferation of NGS testing products in the market, which doubled itself just over the past 2 years, as a result, generating more genomic data. So altogether in 2019, there were 150 petabytes of sequence data generated according to one report by Illumina. And yet this is just the beginning, because there are 10 million people who will be sequenced and their data attached to an EMR in one of 50 population genomics projects that were announced. The ultimate goal in mind, of course, is integrating genomics into a routine standard of care, where clinicians are consulting patient genomics data before making clinical decisions. All that sounds very promising, but while the cost of sequencing has been dropping, the cost of the interpretation of the data is on the rise, to the point it reached nearly 50% of the COGS of a genetic test. Interpretation is now the primary bottleneck in both clinical and research applications, limiting scale and really pushing costs up. I'll refer later to why it is the case, but for now, just assume this is a highly complex research-oriented task, which is done by humans, a small group of highly skilled professionals. So it really is a classic turn to AI sort of problem. The bottom line is that if we are to realize the vision of personalized care, we have to teach machines to do the job of interpretation. Now think about how transformative this is, that machines would understand the clinical implications of our genetics so that it can really be used as decision support by our clinicians in routine care. As a company, this is what we're focusing on, and our talented team of genomic scientists and data scientists are working closely together to identify new ways of interpreting our genomes. We do that because we believe that automated interpretation is a key ingredient to enabling genomic-driven care, and that this is the missing piece for being able to serve mass populations. From a technical perspective, we can now do that thanks to NGS technology that enables us to look further into a patient's genome. We can ask many informative questions about various clinical indications. So as we observe phenotypes in a patient, we can ask, how does genetics influence that? And indeed, evidence of the utility of NGS as frontline diagnosis is being gathered at an increasing pace, looking at various factors such as diagnostic efficacy, change in care management, and overall reduced financial burden. And I laid out here a few examples. For instance, if we were to use exome testing in a child with epilepsy, there's close to 50% likelihood we will identify the genetic cause, often resulting in change of clinical management, including change of treatments in almost 40% of cases. There are over 50 million patients worldwide whose lives can be impacted by this. There are plenty of examples: kidney disease, 700 million patients worldwide, 10% of which is genetic, in 50% of which there is a suggested intervention. The vast majority of them go undiagnosed. Familial hypercholesterolemia, 30 million patients worldwide, 90% of which are undiagnosed. They have 20-fold increased risk of early heart disease. Most of these patients would die at a younger age, while their condition could be managed by simply lowering their LDL levels. And the list goes on. Now, this brings me to the fact there is a huge underserved patient population that could benefit from early diagnosis and intervention. And that's not in a decade. Today, altogether across applications, if we're looking at diagnostics and care of chronic disease, inherited disease, and preventive care, there are billions of people globally who could benefit from genomic-informed care. The studies I've shown were all published in the past few years with a growing body of evidence. So I'm saying it cautiously, but saying it just the same. The clinical guidelines would not be able to overlook new findings that are now revealed at a rapid pace, and they will have to develop a more integrative research clinical approach. Integrating genomics into routine care has been promised for a very long time. So what makes it finally possible? Well, it starts with the digitization of our genome and how cheap it has become. But there's also a convergence of scientific and tech trends that all come into play and make 2020 onward unique. Just over the past few years, we reached an inflection point where the knowledge-based computational biology methods and off-the-shelf algorithmic capabilities have all advanced enough so that when a clinician is seeking to understand the genotype-phenotype connection, technology can surface evidence for the clinician to consider. This is, of course, where companies like ours also benefit from the built-in infrastructure capabilities of AWS. What's important to understand is that over the last decade, genomics has undergone a transformation and really become a data science. And the people that are trying to make clinical meaning of the digital genomic data are called genome analysts. And their primary job is to interpret variants of unknown significance. And since we're still in the early days of deciphering the genome, most variants in most indications are unknown. This new profession of genome analyst is enormously complex. So if in the past a geneticist in a molecular lab was required to have expertise in wet lab processes, today she's mostly seated in front of her computer and needs to understand algorithms, statistical methods, complex data representation. Not only that, but genomic knowledge is actually a moving target with over 1 million new publications published every year. And the information is partly numerical, partly textual, partly visual. So she's processing a lot of data of different types in order to come to a conclusion on every patient. And indeed, analyzing a patient's full genome takes on average 8 hours. With a growing amount of data, this is clearly unsustainable. But if genomics is data science, then just like any other data science domain, it suffers the classical pains: dispersed and unstandardized knowledge, lack of labeled data sets, and the lack of feedback loop to further increase the knowledge base. And this is where we come in. Emedgene provides a robust solution for genomic interpretation at scale. It comes with a necessary tool set to run a high throughput interpretation operation and includes proprietary data sets, knowledge base, and AI applications on top. So essentially an out-of-the-box solution with all the key components health organizations need in order to produce clinical and operational insights from genomic data. Emedgene's AI engine reads structured and unstructured data and holds an up-to-date knowledge base, which is more advanced than what the most thorough geneticist can accomplish by herself. We have evidence within our own Emedgene knowledge graph of 30% more molecular mechanisms than are available via any public database. According to clinical studies we ran with Baylor Genetics, Emedgene's AI engine can also identify clinically significant variants with 98% accuracy and match those with real-world evidence. We also believe AI in healthcare shouldn't be a black box, so we've added models that explain the platform's generated choices with the scientific methodologies that a human can understand. We're lucky to work in close collaboration with fantastic teams from leading research and clinical genomic institutes, and our scientific research division is publishing extensively on our results. As a company, we also work with multiple stakeholders to establish new delivery models and transition existing inefficient budgets to models that are better aligned with positive patient outcomes. Today in particular, we will focus on one such case of the research hospital that demonstrates a new emerging model of in-hospital diagnostics, where the research hospital takes a more active role in diagnosing patients, resulting in improved diagnostic yield. And why is this important? So, according to a study run by Gonzalud et al., on 5 million hospital discharges, approximately 2 to 14% of children that were admitted to a hospital and then discharged had a genetic etiology. And when they were discharged, these kids had a mortality rate that was 5-fold higher versus other discharges. Their hospitalization cost was also higher due to longer stay, bringing the overall burden to the American healthcare economy to, at the very least, $14 billion, but as far as we know, could also be as high as $57 billion, which is nearly 50% of the national bill for pediatric patients. We've presented here today a fascinating case study of Boston Children's Hospital, which is taking a proactive approach of integrating research findings into clinical settings. I find it to be particularly exciting, as over the course of just a little over 1 year, they were able to set up the end-to-end infrastructure required to support the integration of research insights into clinical care. This is unprecedented and was hard to achieve even if they were to start only 3 years earlier. In a recent symposium, clinicians refer to this new workflow as transformative. So I'm going to hand it over to Dr. Shira Rockowitz, data science lead at Children's, who will share more about the work they've done internally and how they worked over the past year or so with Emedgene for that purpose. Shira?
Thank you, Einat. Hi, my name is Shira Rockowitz. I'm the data science lead in research computing at Boston Children's Hospital, and I'll be describing today the Children's Rare Disease Cohorts, an integrative research and clinical genomics initiative. I'll be showing how our institution has worked systematically to integrate clinical and research efforts to provide cutting-edge genomic insights for our patients. The initiative was developed to provide large-scale sequencing to rare disease cohort patients, the vision being that this kind of sequencing would drive therapeutics and research discovery, push forward the delivery of the best standard of care, and lead to the development of collaborative networks. The Children's Rare Disease Cohorts initiative was internally funded. In the first year, we set up the initiative, sequenced over 2,000 patients and family members, and preliminary analyses have identified hundreds of variants of interest. Into year 2, we brought that number of patients sequenced close to 6,000, rapidly onboarded COVID and other cohorts that are pushing forward with advanced analytics. To do this, we needed to accomplish a number of complex tasks. First, we needed to align stakeholders across the institution and spur cultural changes that enable data to be shared and used. What I'm going to share in this talk is working. Investigators across the institution are reporting positive outcomes resulting from this work. Today, we have in place a database with rare patient data, clinician scientists that are leading in these research areas, and a system to bridge the data and expertise that can easily be extended. I want to highlight that this work has really been cross-institutional and included major contributions from cohort study PIs, their research teams, leadership, patient consenting, cohort selection, genomics, and project execution experts. Though I'll be describing work that's happened within the last 1.5 years, this project has deep roots. Researchers at Boston Children's Hospital have been collecting and analyzing genomics data for decades. Internal stakeholders developed specific recommendations to leadership, and a cohort subcommittee was assembled and conducted a survey across the institution to ascertain what the need for sequencing was across the many departments and divisions. The response was overwhelming, with Boston Children's Hospital investigators projecting that over 25,000 patients would benefit from sequencing. 2 cohorts that had high genetic contributions to the disease and high ongoing enrollment efforts were selected from the survey. The project was funded in 2018, late in 2018, and there were a lot of alignments in setting up this initiative. The initiative was really data-driven, with many bottlenecks being identified as we started collecting data, and many optimizations to workflows and infrastructure made in response to bottlenecks. In principle, this execution timeline I'm showing here looks really straightforward, but the simple timeline really represents complex alignments across the institution that included cultural changes. The support provided by the initiative facilitated the implementation of the alignments. Phase 1 of the project really focused on setting up the mechanisms for this initiative. And as we start phase 2, we're looking forward to new collaborations, cohorts, technologies, and data sharing. Our paper describing phase 1 of the project has been accepted in Nature Genomic Medicine. Today, close to 6,000 patients have been enrolled in Sequence. Each of these cohorts that were selected had high evidence of genetic contributions to disease, a potential for funding to make a difference in the project, and capacity to support the recruitment and analyses. Each cohort is led by a clinician scientist with expertise in a specific disease. This table shows the disease focus in each cohort, an estimate of the total BCH patient population, and the number of samples collected thus far from enrolled patients. I want to highlight first that this represents a huge amount of work on the part of each of the participating research groups. By implementing this process that leverages existing expertise, we were able to rapidly scale up consenting, and all of these patients were consented and sequenced in the last year and a half. Also, we've been able to rapidly accommodate new cohorts and workflows, as evidenced by the addition of a COVID cohort in the last few months. As part of the initiative, we've streamlined a lot of administrative processes, including aligning consenting around data sharing, support for therapeutics development, and broad rather than disease-specific use to really enable data harmonization. Process alignments have been led by practicality and the application of technical innovations, particularly in the arenas of patient enrollment and sample collection. Finally, technical alignments around integrating data that has been historically siloed. Last but not least, there was clinical alignment. One of the main things to align with was to align with the clinical needs of the hospital and follow-up that would potentially benefit the patient. Samples were sent for clinical clear grade sequencing. Data is returned to our research environment and loaded into Boston Children's Hospital Genomic Learning System for analysis. Research teams identify variants of interest that might explain a patient's phenotype and work with the patient's clinician to have the variant clinically confirmed by the clinical sequencing company using the original sample collected. Information from the clinical sequencing that is done at the hospital is also included in the genomic learning system. Patients are ending up with clinical confirmations and new genetic diagnoses. Clinicians are able to leverage this genomic learning system that contains feedback from research and clinical testing, and researchers are able to help their patients get access to cutting-edge diagnoses, altogether improving our ability to provide genomic insights to our patients. With the alignment of the consenting principles, data can be shared, but there's frequently a gap between that theory and practice. Integration of data originating from different data sources is a complex task. To solve this problem, we created a genomic learning system to uniformly process and analyze patient data, with the idea that this would accelerate discovery and foster collaboration. By deploying this across the institution, any researcher, regardless of their bioinformatics background, can start analyzing patient data immediately. The genomic learning system is a centralized data repository where harmonized data is generated and stored. Having a single data store itself provides functionality by providing data to other systems rather than replicating data multiple times. The genomic learning system is modular and has robust analytics platforms, which can answer many questions and which we keep expanding. It's worth noting that the genomic learning system is not a single interface, but many interfaces in a single project, all working jointly, that we have worked and continue to work with various groups to develop. We're also prioritizing security in this implementation. Together, the genomic learning system is extensible, flexible, and integrated. Much of this infrastructure was set up within the first year of the project, and we were able to do this so quickly while maintaining a lean in-house team of bioinformaticians because we worked with multiple commercial vendors to innovate. We found that carefully reviewed use of commercial vendors accelerated the use of cutting-edge technologies, mitigated costs, and allowed us to rapidly meet the requirements specific to Boston Children's Hospital researchers and processes, and enable our research teams to remain focused around their leading expertise in rare diseases and discovery science. One of the systems that was integrated with the genomic learning system was Emedgene, which provided Boston Children's Hospital researchers with a mechanism to rapidly prioritize variants. Any individual's whole exome sequencing data will contain thousands of variants. So how do you pick the one that's causal for a patient's disease? This process is complex, and analysis of each variant is time-consuming and expertise requiring. With Emedgene's knowledge graph and variant prioritization algorithms, researchers were able to leverage up-to-date information from the ever-growing literature and start their analyses with a shorter list of machine learning prioritized variants. And they can also see why a specific variant has been prioritized, how they match a patient's phenotype, and so as to more rapidly assess whether or not the specific variant may be causal for the patient's disease. It's early days for the initiative, and many analyses are still ongoing. As such, we expect that many more results as time progresses. Nevertheless, we were able to identify hundreds of variants of interest, many of these variants of interest having known gene-disease-drug relationships or clinical trials, suggesting that there might be meaningful clinical interventions. Research analyses led to the clinical confirmation of variants in many patients, some patients having atypical or mild presentations that would not have typically received clinical sequencing as a diagnostic for their disease. Other patients were connected with services. For example, one family was connected to a clinical trial at a nearby hospital for which they were eligible based on their genetic diagnosis, and another enabled a patient with an atypical presentation to be seen at a specialized multidisciplinary clinic. We've provided a model for institutional integration of data to support both discovery and clinical use. This work shows that there's value in aligning consent, developing a modular genomic learning system, and integrating clinical and research data sources, and that being able to do this depends on the contribution of many stakeholders. The first phase of the project established the collection, integration, and sharing of research, clinical, and genomic data within the genomic learning system. In the next phase, the focus will shift to data analysis, integration with other institutions, and integration of other modalities. The approaches and lessons from our work serve as an example for developing institutional workflows for the integrated analysis of research, clinical, and genomic data. Adoption of comparable strategies by other institutions will further enhance research and clinical collaborations, provide greater power for genomic analyses, and accelerate pediatric precision medicine. This project, a project of this breadth, has only been possible with the enthusiastic collaboration by leadership, numerous BCH investigators, and their respective research teams. Their vision, input, and feedback on every aspect of the children's rare disease cohorts were invaluable in developing integrated workflows across our institutions to meet the needs of patients, researchers, and clinicians. And the first phase of this manuscript describing the first year of the initiative has been accepted in Nature Genomic Medicine. Handing it back to Einat.
Thank you, Shira, and the wonderful researchers at Children's for sharing the inspiring work you do with us. It's really great to be a part of it. What I find transformative in the use case shared here today is that it serves as a proof of concept to a new delivery model, often in-hospital diagnostics, where the research hospital takes a more proactive role in diagnosing patients, regardless of whether it has an internal clinical lab or works with a reference lab, and by that accelerating research and potentially improving patient outcomes. As a company, we provide technologies to enable the rapid implementation of such a framework, make it easy to launch and operate a high throughput interpretation operation. And by that, we're reducing the cost and enabling to quickly scale this framework to thousands of patients. Most important is that we believe that AI-based genomic interpretation can truly unlock a new paradigm of care and drive efficiencies and cost reduction across the entire continuum of stakeholders. If you're a hospital CIO looking to improve patients' outcomes, or an innovative officer looking to drive efficiency in large-scale genomic operation, we welcome you to come and speak to us. Thanks everyone for listening.